Thank you very much for the invitation. And I just congratulating to this symposium. I'm sorry that I cannot come to participate. I did participate last one, but this pandemic prevent us to really, mobile. So I do this online. Hopefully, can achieve the effect. Next slides. So relaxin family of peptides, right? This is actually number 4. So GPCR are well characterized drug target with significant therapeutic value. So of the marketed drugs, over 33%, between 33 to 34%, are actually targeting G protein coupled receptors. Now, this particular receptor we call relaxin family peptide receptor 4, also known as RXFP4, previous called GPCR 142 or GPR 100. They are all the same. So it's a class A GPCR, which was discovered about 17 years ago through the human genome science project. Next slide. So this receptor is very interesting. So widely distributed from the brain all the way to different organs all over the body. So mostly importantly is strikingly also is the ligand itself used to be an orphan receptor. Later was discovered its endogenous ligand is called insulin-like peptide 5. We call it INSU5. So this peptide is very complicated. Like insulin has A chain, B chain. Within the molecule, there are 4 disulfide found. So because of this very complex structure, it is very difficult to synthesize or even through genetic means to make it. So only a couple of labs in the world were able to make it. But now it's kind of commercially available, but it's still very expensive. Now, relaxin family peptides are their receptors. There are different receptors. The most importantly is this receptor 3 and the receptor 4. So they are, in terms of receptor 4, which is our topic today, are mainly signaling pathways through G protein. So it has effect on CAMP and also on ERK, as well as to less extent signal the calcium mobilization. Next slide. Early studies either for receptor knockout or ligand knockout mouse also both shows some dysfunction in glucose as well as lipid metabolism. So about 5 years ago, we published a paper. It indicates that this particular peptide, insulin-like peptide 5, promotes the release of insulin and GLP-1. GLP-1 is actually a glucagon-like peptide 1. It is also class B GPCR ligand. And this insulin-like peptide 5 also increases glucose tolerance in normal and DBDB mice. Now later, people find that insulin-5 also enhances the food intake. So its action is very complicated. In the meanwhile, it has some anti-diabetic effect, but on the other hand, it increases food intake. It's just like insulin. Next slide. So there is so far most ligand studied are peptides. For example, one of the 4 agonists called insulin 5-A13 may promote show some effect on treating constipation. So because it also slowing the colonic proportion. So that means there's some effect on the gut as well as colon. Another analog called insulin-like peptide 5 R27S is associated with increased insulin sensitivity and weight loss. Recently, people find that insulin-like peptide 5 are significantly increased the value level in diabetic patients, suggesting its role, insulin secretion in relationship with compensatory role of insulin secretion. But anyway, this peptide itself is very much like insulin. And in that sense, but the problem is that it is very, next slides. It is very difficult to make this peptide. So people are developing various assays or ligands, but all of them are what do we call it? Peptide based. None of them are small molecule based. But if you see the small molecule on the right bottom part of compound 1, 2, 3, these are agonists, but they are not selective. They both act on RXFP3 as well as RXFP4. So it's there's no receptor selectivity, so cannot be used for drug purposes. Next slide. So that is our effort to launch a high-throughput screening using our compound library in Shanghai. The assay itself is we use just a CAMP assay, and we optimized cell density and force curling concentration. Force curling is a control compound. It stimulates CAMP accumulation. So then also we use human insulin like peptide 5, as well as this R3-I5. So that's actually peptide, but as I said, it's a dual agonist on both receptor 3 and receptor 4. So the assay itself is okay, has a Z factor over 0.6, and signal to background noise is 2.33. So it's good enough for us to perform high throughput screening assay. So in fact, we performed a high screen campaign against 52,000 small molecule compound, all synthetic compound. Next slide. Of which we find 109 initial hits, that's primary hits. Then we counter screen again. Next slide. In the end, we only find 3 active compounds. They are displayed here. So these compounds are compared to native peptides, their potency is not very high. And we concentrated on this compound, the first compound, called JK0621, that compound, to further our studies. So this compound actually is a racemic mixture. So that means one's S form, one's R form. Next slides. So we actually use chemical method to isolate this the mixture. Called one is JK1. The other actually is JK2. So we use the efficacy actually lies with JK1, not JK2. I think so. That's why we used this assay to further our studies in vitro. So from these graphs, you can see, this JK2 has no effect, while JK1 representing the most activity of this mixture. Then we checked them about this CMP effect, you can see JK1 has similar effect as the control compound, control peptides or compound, but the potent is about 2 logs less, you know. And we also checked this compound in mouse IXFP4 receptor and showed a similar effect. And this effect, if you block the acid bioptesis toxin, which actually block the G-alpha IO protein, that will inhibit the CMP response. Next slide. So we further checked, you know, these activity. For example, we checked this in the RXFP3 cells. You don't see any effect on JK1. But if you check on 5-HT1B receptor, you do not see JK1 effect as well. And if it's blank cells, you see nothing on these both receptors. But receptor, if we use a reporting assay, which is CRE, response element driven assay, again, you can see the response for RXFP4, but not in RXFP3. Only until the last very high concentration, you see some drop. Probably this, as I show later, is due to toxicity when you get very high concentration. Again, not in the parental blank cells. Next slide. Then we study this receptor specificity in terms of secondary pathway. For its sister receptor, RXFP3, you can see, you know, the only the dual agonist has effect, but JK1 does not. On ERK, and as well as on the you can see relaxin 3 effect, but you cannot see this JK1 effect. We also checked this assay on beta-arrestin 1 and arrest 2, and this compound actually, just like a peptide itself, the effect on beta-arrestin recruitment of both 1 and 2 are very minimal. Next. So in terms of ERK phosphorylation assay, you still can see this compound act like a native peptide, insulin-like 5, as well as dual agonist, but its potency is much lower. And this effect actually can be blocked by Merck inhibitor, the mitogen activated protein kinase inhibitor that inhibits the ERK response. And you can see these, you know, in both situations, you can see this forskolin, you know, CMP can also be CMP. If you use the right panel, if you look at the assay that potassium toxin actually inhibited the reaction. So calcium mobilization, it also has the similar effect. The potency, again, is lower. And in the blank cells, you don't see any effect on these cells. So next slide. So we also use this to check beta-arrestin recruitment in the RXFP4 environment. But in both situations, this pathway is not significant, just like in RXFP3, it's very minimal. And the receptor affinity, as I showed you, the left bottom panel, actually is not, if you see, compared to R3, I5, this dual agonist on this RXP4, this compound JK1 is much potent than the other. And in high concentrations, the compound has some toxicity. So basically, we use this compound as template, are making analogs and try to do structure-based drug designing and to improve, optimize the compound potency. Now, so far, we have one paper actually submitted with some new compound that is better than JK1, but it's not strikingly improving the potency, because you know that the ligand itself, the native ligand, the insulin-like peptide 5, which is a very large molecule. Such a large molecule occupy this receptor. Try to use a small molecule as agonist. It can never be able to elicit similar response in the same range, potency and efficacy. So we actually now, because my group also doing structure biology studies. So now we are making construct for both RXFP3 and RXFP4, and try to determine the structure of these compounds with their native ligand, which is one's relaxin, the other one's insulin-like peptide 5. And in complex, again, also with a small molecule, if we can, we do not know yet. And once we understand the structure information, that probably will help us to design better compound and also understand how this small molecule can resemble a large peptide in terms of this in vitro activity. So we have not done any in vivo study yet, because this compound itself it requires this separation. That is quite expensive. So we are looking for some better compound, so that we can, again, we need to separate them to accumulate enough quantity to do some in vivo studies. Next slides. With this, let me conclude this very short presentation, and I welcome to any questions.
Thank you very much for your nice presentation. Any question?
So, Dr. Wang?
Yes.
So we do not have any questions from the audience.
Yes.
But participants of the symposium have submitted some questions in advance. So I'd like to read those questions to you.
Okay.
So question number 1. So what is your strategy, or do you have any methods for selecting compounds for screening among a tremendous number of chemical libraries?
So I think in terms of what we do, because we have a collection of 2.2 million compounds. All of them are purified and synthetic compounds. So what we did is we have sub-libraries. For example, GPCR library. So these are compounds which the structure is very much resemble what the GPCR ligand reported in the literature. And we interpret this in a very broad sense. So these are one collection. So we call it a GPCR collection, sub-library. And also we have kinase library. So for GPCR screening, what we do is sometimes we go to GPCR sub-library to start screening. But in this case, not. Why? Let me tell you why. Because GPCR library, their compound very much, you will get hit. But the structure itself are very much like those drugs on the market, or these drugs in the clinical trial or failed, but they are as a chemical probe for GPCR studies, or tool drugs. So in this case, we have a diverse set That means we have a collection about 60,000 compounds, which are representing different classes of compound in terms of structure. So we pull them together. So we start screening, but not like 52,000 at one time. So we go about 8,000 at each run. So if you see something, then you carry on. If you see something, you carry on. So that's the way. I think initial screening, if you want to find some new compound that maybe help you eventually generate intellectual properties, you have to go through this very diverse set in order to see all the possible chemical information.
So I'm not sure whether we have time to ask Mr. Wang to answer the second question. So let me ask you just one last question. So this is the last question for you, Dr. Wang. Can you briefly explain the current status of the Chinese chemical bank?
Yes, so our compound library is located in Shanghai. And the Chinese government support this library through us. Then we have about 12, we call the satellite libraries around China. So these are in Beijing, Nanjing, Hangzhou, Tianjin, Guangzhou, different cities. They are mainly located either in Chinese Academy of Sciences or Academy of Medical Sciences and universities, such as Peking University, Nankai University, or Zhongshan University, Zhejiang University. So we have a jointly apply grant or jointly apply for government support. So through this mechanism, we can physically, the library, each set library, they are maintained in their own way, but it's all standardized. We can call their compound if we need. So we have done recently for COVID-19, right? COVID-19. We did a real screening over 1.8 million compounds and find some very interesting drug hits. So it's a really running capacity. We do about 50 campaigns, HTS campaign a year. I mean, these targets are mainly come from different universities, scientists. They bring their assay to Shanghai. Or sometimes if they have their own facilities good enough, we send a compound for them to screening. So overall, around 50 campaigns per year.
So Dr. Wang, thank you very much for your explanation, and thank you very much for joining us today.
Thank you very much. Thank you. Congratulations again for this symposium.